All right, let's go over this here, guys. All right, so we start with number 2. Number 2 is the smooth ER. Okay, I will accept any one of the following functions for smooth ER. Breaks down complex carbohydrates, repairs membranes, makes lipids, detoxifies poisons, makes hormones. They have any one of those, they can have the mark for the function. Okay, so 1 mark for the name, 1 mark for the function. All right, number 3 is pointing at the nucleus. The nucleus stores DNA. Okay. If they wrote brain of the cell, give them a 0 right now. You can stop marking. I'm just kidding. Okay. We're looking at number 5 now. Number 5 is the outside of the nucleus, the nuclear membrane. Okay, and the nuclear membrane keeps stuff out of the nucleus. Okay, if they have something like protects the DNA, I suppose I could accept that. Okay, but essentially it's just a barrier. Make sure nothing gets into the nucleus. Okay. Okay.
Allows DNA and RNA flushed out of the nucleus.
Good. Yep. Yep. Any others? All right. For number 6, the little dots are the ribosomes. They make protein, or synthesize protein, if they wrote it technically. Number 7 is the Golgi apparatus. Okay, it excretes wastes, gets rid of wastes, makes transport vesicles, okay, something along those lines. Okay, questions on the Golgi? All right, number 8 is the cell membrane. What 2 words am I looking for here? Endocytosis and exocytosis. Okay, number 9 is the mitochondria. The mitochondria carries out cellular respiration or burns sugar for energy. Either one of those would be fine. No, not too simple. Okay, number 10 is the lysosome. Okay, and the lysosome digests food, breaks down food particles, okay, something along those lines. Contains digestive enzymes, I guess I would take too, okay, even though that's technically more the peroxisome. Okay, number 12, the stringy things. Hopefully they didn't leave that as the name. Okay, we're looking for cytoskeleton here. Okay, but I'll accept microfibers or microfilaments as well. Okay, and their job is to support the cell. Keep the shape of the cell. Keep cell from rupturing during a shape change. Anything about shape. Okay. Okay, and number 11. I know we're kind of skipping around here, but okay, number 11 is the centrioles. And if they have anything about helping with cell division or making sure DNA gets to the right side. Oh, okay. Okay. They help with cell division or make sure DNA gets to the right places during cell division. Okay. All right, it's out of 20. Give them a mark out of 20. Let them see it. Then bring them and put them on this desk behind Blake. Did everyone get that today? The desk behind Blake, please.
Would ribose be good for ribosomes?
No, they got to have the whole, yeah. Well, the other day we talked about the big 3. Okay, these are the big 3 methods of cellular transport. Osmosis, which moves water across the membrane to balance salt. Diffusion, which is intracellular transport. Things move from high concentration to low concentration within the cell. And active transport, which pumps material across the membrane against the gradient. We are concerned with diffusion today because diffusion is why cells don't get overly large. Okay, diffusion is slow. It's not targeted. Okay, and only works well over small distances. That is what limits the upper size of a cell. Okay, a cell gets too big, diffusion will not get material where it needs to go effectively. Okay, so the problem we're investigating, okay, how does the surface area relate to the volume in cells of different sizes? Does dividing a large cell into smaller cells affect the surface area to volume ratio? Okay, I haven't used those terms before, surface area to volume. I've always just talked about the size of the cell. Well, surface area to volume ratio is essentially a measurement of cell size. Okay, as a cell increases in size, its surface area increases and its volume increases. And I showed you this earlier in the week, okay, when I went over this. You guys remember that? Okay, and I showed you, here's a cube that's 10 by 10 by 10, and here's a cube that's 20 by 20 by 20. You guys remember me showing you that? Because I didn't show you guys, I showed periods 2 and 3. Okay. Okay, we're going to do the math on here. It'll be fine. We're actually going to do the math together and you'll get it. Okay, so surface area to volume ratio simply talks about if I have a small cell, like this cube here labeled micro, okay, it has a small surface area and a small volume. And its surface area is actually greater than its volume. That's a good situation. If a cell has a large surface area and a small volume, it's going to be able to absorb a lot of material through its membrane, and it doesn't have a big volume for diffusion to move that stuff it absorbs through. That's a good situation. The bigger a cell gets, the bigger its volume gets. And volume increases faster than surface area, because surface area is two-dimensional, where volume is three-dimensional. Okay, so if I want to calculate the surface area of a cube, I take the length of the cube, multiply it by the width of the cube, okay, and then multiply by the number of sides. Okay, that gives me the surface area, because that gives me the area of one side, and all the sides are a square, so I multiply by 6. Everyone okay with that? Okay, to calculate the volume of this, I go length times width times height. Okay, now, if I have a large single-celled organism, that's what macro represents, is a very large single-celled organism, and I compare it to this would represent a what? A multicellular organism. It's the same size. It's got the same volume. Does it have the same surface area? No, it doesn't. Okay? This large single cell has only the surface area of the outside of the box. This has the surface area of the outside of every box. And there are 1,000 boxes in here. It's 10 by 10 by 10. Okay? Does becoming multicellular help you to overcome a surface area and volume problem? Yeah. Okay? What we're going to find here is that the surface area and volume of micro and the surface area volume ratio of multimicro are the same. Okay? The reason for that? Well, what's multimicro made up of? A whole bunch of micros. Okay? Each one with a good surface area to volume ratio. Whereas if you're a single large cell, you've got all this volume and only the outer surface to feed it. Okay? Is everyone following me on that? Okay. So your hypothesis, if and then statement that talks about transport by diffusion. So generate a hypothesis about the relationship between surface area and volume in cells and how that can affect the functioning of the cell. So I would say the if part should be about this. Okay? If cells transport materials by diffusion. There you go. There's the if part. Okay? Everybody writes that down. Everybody gets the if part right. Okay? The and part should talk about what we just finished talking about, which is the effect of size, okay, on volume and surface area. So, and as a cell increases in size, this happens. Okay? That should be the and part. The then part I'm going to leave to you. Okay? But you should mention something about multicellularity and being unicellular and who has a bigger advantage in terms of cell transport. I'm hoping we're going to all have it done at the end of class because we're going to do a bunch of it together here. Okay. Remember, there's not going to be any evaluation. Okay. There's no design. Right. There's no design part. You're going to have a hypothesis. The calculations we're doing together, and you guys got to answer the analysis, come up with a conclusion. Okay. Should be able to do that in the time we have. I have set the due date as tomorrow at noon. Okay. But I think you probably will get it done. Okay. Okay. So you've got a chart like this in yours. Okay, already built for you. Right? I'm going to use my wonderful hand-drawn one here. Okay, what we're doing is we are calculating the surface area and volume, and then the surface area to volume ratio of the 3 cubes. All right, since I didn't tell you to bring a calculator today, we're just going to go through it together. All right, so if I want to calculate the surface area of micro, I'm going to take its length, 1, its width, 1, and multiply by the number of sides, 6. Okay, so I'm going to find then that its surface area is 1 times 1 times 6, which is 6. Okay, so we're going to say 6 micrometers square, because it's surface area and surface area is square. Oh, just put U. Yeah, just put a U. I mean, you can insert symbol and look from U, but that takes too long. Okay, just put U and I'll know what you mean. Okay, to calculate the volume of micro, length times width times height, 1 times 1 times 1. I'm pretty sure I can do that one in my head. I'm pretty sure it's 1. Okay, 1 cubic micrometer. Okay, so the ratio then of surface area to volume is how much? 6 to 1. Anytime surface area is greater than volume, the cell is in good shape. Even if that ratio is only 2 to 1. I mean, that's not great, but it's okay as long as surface area is the greater of the numbers. All right, let's look at macro. Macro is 10 times bigger on one side than micro is. So it's a very large single cell. So to calculate its surface area, 10 times 10 is 100, times 6, 600. All right, so it has a surface area of 600 micrometers. And it has a volume of 10 times 10 times 10, which is 1,000 cubic micrometers. So our ratio is no longer 6 to 1, it's now, or 0.6 to 1, okay? Or 6 to 10, yeah, but let's write it as the volume always ends up being 1, okay? So, is my surface area a lot smaller than my volume? Okay, so we're not looking at a positive situation here for a large single-celled organism. All right, and then for multi-micro, okay? Multi-micro is made up of how many micros? 1,000, okay? Because there's 10 micros on a side. The surface area of micro was 6 micrometers square. So what's the surface area of multi-micro? 6,000. Okay, its volume is the same as multi-micro, or sorry, as macro. It's the same because it's 10 by 10 by 10. Okay, it doesn't change the volume. It's still 1,000 micrometers, cubic micrometers. What's our surface area to volume ratio now? 6 to 1. That should make sense. If I'm made up of a whole bunch of small cells and they all have a surface area to volume ratio of 6 to 1, my overall surface area to volume ratio should be the same, 6 to 1. Okay. Yesterday we talked about multicellularity and why it was important. Here's why it's important. It overcomes the problems a single-celled organism encounters when it becomes large. Okay? Multicellularity allowed organisms to become large without having transport problems. Okay? All right. That's 5 marks right there. You're welcome. Okay? Because it's on your thing there. Okay. So you got your analysis and interpretation here. So I'm just going to quickly go through that so there's no confusion. Okay? So when the cube dimensions increase from 1 to 10, that is from micro to macro. Okay, so when we increase the size from micro to macro, the increase is a factor of 10. That is, one side of macro is 10 times bigger than one side of micro. Okay? What factor of increase is there for the surface area? Okay? So in other words, how many times bigger is the surface area of macro than micro? And then same for the volume. How many times bigger is macro's volume than micro's volume? Okay? So you're just looking at two numbers here. Okay? For that question. Okay? For the two large cubes, for number 2, okay? The size and the volume have a factor of 1. That's the hard way of saying they're the same. Okay? They have the same volume and they're the same size. But there's a difference in the surface area. How many times bigger is the surface area of multi-micro than macro? Okay? That's what number 2 is asking. Okay? Question number 3. For which two cubes does the ratio between volume and surface area change when their size increases? So if we go back to our cubes here, if I make micro bigger, it essentially becomes macro. Okay? Does its surface area to volume ratio change when that happens? If I make macro bigger, does its surface area to volume ratio change? If I make multi-micro bigger, we have to assume that if I make multi-micro bigger, it's going to fill up with more micros. Will its surface area to volume ratio change? That's the key, is assume that when multi-micro gets larger, it is still filled up with little micros. Okay? When you're answering question number 3. Okay? Number 4, use the diagram below right to answer this question. Okay? So I've put the measurements on here, because obviously you can't measure on the screen. Okay? So I've put the measurements of these boxes on here. Okay? What you will find is this. When you calculate their surface area and their volumes are all very close. Okay? All 3 of those boxes have a very similar volume. What we want to find out is, by changing their shape, did we change their surface area to volume ratio? So what you need to do is find the surface area and volume of each of these 3 boxes, calculate their surface area to volume ratios, and come up with a conclusion about shape and its effect on the surface area to volume ratio. Okay? Now, remember, A is a cube. It doesn't quite look like it, but it is. It's 10 by 10 by 10. Okay? When you're calculating the surface area of an irregular object, okay, you have to calculate each side and then add them up. Okay? But remember that this side and the back side are the same. So if you calculate the front, multiply it by 2. Okay? Calculate the top, multiply it by 2 because it's the same as the bottom. Calculate this side, multiply it by 2 because it's the same as the other side. Everybody follow there? Yes? Okay? And then same thing for box C. That won't take you very long, and you should be able to use an online calculator, okay, in order to do that. Okay, number 5 is basically asking a similar question. Okay, number 5 is looking at 3 different kinds of cells in your body. A liver cell, which is like box A, a skin cell, which is like box B, and a nerve cell, which is like box C. Okay, so it's asking, okay, how have the cells adapted to deal with the problems of surface area to volume ratios? Okay, remember, a liver cell is in your liver. Okay, the liver has more blood in it than any other organ in your body. Okay, at any one time. Okay, your skin cells are obviously exposed to the outside environment on one side and are only fed on the other. Okay, your nerve cells, some of them, the ones in your spine, are over 1 meter long. Okay, they're over 1 meter long, but are they big in all dimensions? Okay, something to think about when you're answering that question. Okay, and then number 6 is asking about the shape of red blood cells, okay, and how that shape is beneficial for carrying oxygen. Okay, and then number 7 is probably the most important question in the analysis. Most materials get from place to place in the cell by diffusion. What effect would a low surface area to volume ratio have on intracellular transport? Oh, analysis is always out of 5. Yeah, I don't, it's not 1 mark per question, it's 5 marks in general. I go through your questions and I give you a mark in general out of 5. Oh yeah, but you don't have to. Yeah, okay, we're done that. We've already did that. Okay, and then conclusion, okay, restate your hypothesis and tell me, based on what you saw here with these cubes, the multi-micro and macro and micro, okay, what did you learn about surface area, cell size, and transport? Okay, and that's it. You don't have any evaluation because there are no sources of error in this lab. Okay, so there's no design, no evaluation, and we've done all the math already. Well, except for this little bit here, which won't take you very long. Okay, you still have a half an hour. Okay. So do your best to get finished in class, but if you don't, okay, we'll say tomorrow at noon will be the deadline for these. Okay? But I don't think you should have too much trouble getting done in class.